Now we're getting into the production of viral nucleic acids and proteins. This is based on the Baltimore classification scheme, which came out just before 1975, and Baltimore's group shared the 1975 Nobel Prize for their development. It's a very elegant classification scheme, and it's based on the relationship of the viral nucleic acids to messenger RNA. The Baltimore classification scheme is broken down into a number of different classes. So class 1 viruses are double-stranded DNA viruses. Class 2 are single-stranded DNA viruses. Class 3 are double-stranded RNA viruses. Class 4 and 5 are single-stranded RNA viruses in the plus or minus configuration. More on that in a minute. Class 6 viruses tend to be retroviruses, and Class 7 are the double-stranded viruses that replicate through an RNA intermediate. And unfortunately, we don't have time to go through Class 7. Class 7 are another type of retrovirus. This figure shows how viruses work. The ultimate goal is the production of messenger RNA. As we can see here, messenger RNA has, in its normal usage, a plus configuration. That's just by convention. And that allows us to understand how these viruses can actually work. So let's deal with the DNA viruses. They're very simple, very straightforward. Why? Because they essentially use your host machinery to function. So double-stranded DNA viruses, class 1 and class 7, essentially use your machinery to produce messenger RNA. Single-stranded plus strand DNA viruses work through a double-stranded intermediate using your host machinery and eventually produce messenger RNA by your normal cell's processes. RNA viruses are a little bit different. Why? Because we actually do not make the RNA-dependent RNA polymerase that viruses need to function. We don't make it. So how do they do that? Ultimately, they want to make messenger RNA too. So let's look at the easiest case, and that is the class 4 single-stranded RNA plus configuration. This virus, as soon as it gets into its host cell, essentially mimics messenger RNA. Why? Because it's in the plus configuration. So this viral single-stranded RNA is essentially the same as the messenger RNA. As soon as it gets in, it starts to produce early phase proteins, specifically its own RNA-dependent RNA polymerase. Double-stranded RNA and single-stranded RNA negative configuration, negative polarity, essentially has a little bit of an issue. Why? Because in terms of the single-stranded negative polarity, it doesn't behave like RNA, messenger RNA. Nor does the double-stranded RNA virus in the plus or minus configuration. You would think that as soon as it gets into the cell, this double-stranded RNA would separate and would be able to proceed as normal, the plus strand being automatically used as messenger RNA. But that's not the case. Why? Because it's probably not energetically favorable for that virus to separate on its own. It needs an enzyme to do that. So with this virus, double-stranded RNA viruses and single-stranded RNA viruses negative polarity, what they have to do is bring along within their virion particle a small amount of that RNA-dependent RNA polymerase in order to function. And we'll see that in a minute. In addition, we have the retroviruses out here, which uses a very specialized enzyme we call reverse transcriptase that produces a double-stranded DNA intermediate. Here we have we're going to deal with this one here. This is the positive sense strand, single-stranded virus. So it comes in. Here we have the single-stranded RNA plus configuration. Immediately we have produced the early phase proteins, and this is going to be the polymerase. And the RNA-dependent RNA polymerase will be used to manufacture a complementary strand. And so the virus is going to make many of these because it has to make many positive strands in order for it to make more copies of itself for final release. So here we have a scenario where we're making complementary strands from the positive strand, making negative strands, and from the negative strands that can be used as a template to make more positive strands, and those positive strands are going to do two things. One, they're going to make more viral protein, and two, they're going to be used in terms of the assembly phase, and then once all of this has been assembled, then the virus can leave the actual host. And within that host, you have copies of the nucleic acid in the plus configuration, single-stranded RNA. Let's look at the single-stranded virus that has a negative polarity to it. Here, it has to bring along with it its own RNA-dependent RNA polymerase in order to function. So once it has in the cell, and when it has its own RNA-dependent RNA polymerase, it can actually start to make more strands, the complementary strands. So it's going to make many copies of this plus single-stranded RNA. And this will now be able to make viral proteins, because now it's gone from the negative to the plus configuration by using its RNA-dependent RNA polymerase. And we have a scenario now where we have lots of messenger RNA now being produced, which is a viral RNA at this point. Early phase proteins will be manufactured, in other words, more polymerase proteins will be made, and late phase proteins will also be manufactured down the road. And from here, we make many copies of the negative strand. And this negative strand will be grouped with the viral proteins to make a mature virion during assembly phase. When the virus leaves the host, it has within its virion a copy of the nucleic acid. In this particular case, it's going to be the negative strand, single-stranded RNA, plus it has to have other components that are necessary for its survival. And in this case, it has to bring along with it RNA-dependent RNA polymerase. And a similar situation happens with the double-stranded RNA virus. It has both configurations, plus and minus, but it doesn't function on its own. It needs that RNA-dependent RNA polymerase in order to produce more copies of itself. So it produces more copies of the negative strand and more copies of the positive strand. The positive strand will act as messenger RNA, produce early phase and late phase viral proteins, and in terms of the assembly stage, these will come together and produce a mature virion. In terms of the mature virion, this particular virus has to have both strands, so we have both negative and positive strands coming together. In addition, it has to have components that are necessary for the viral survival. So in this case, it has to bring along within the virion itself copies of that RNA-dependent RNA polymerase. And when it leaves the host to go off to infect, it's going to envelope out and infect other hosts. And when it enters the other host, it will start exactly the same way. So it'll be a binding to the host cell, releasing nucleic acids, the RNA-dependent RNA polymerase goes to work, and the whole cycle goes on and on and on. The next few slides essentially summarize what we've been talking about through the Baltimore classification. So once the host has been infected, new copies of the viral genome are made. And the generation of messenger RNA occurs first. And so it depends on the nature of that specific virus. Okay? And this virus genome serves as the template for that viral messenger RNA. In the case of a single-stranded plus strand RNA viruses, the viral RNA itself is the messenger RNA. That only happens in that one specific class, class 4. The other two have to bring along the essential transcriptional enzymes that are necessary for the virion to be functional. So within that virion particle, you will have not only the nucleic acid, but you will have, in this particular case, it's going to be RNA, negative polarity, or the double-stranded RNA. You will have to have copies of that RNA-dependent RNA polymerase in order for the virion to be functional. If, for some reason, when the virus matures and that RNA-dependent RNA polymerase is not there, then that virion is not viable. So in terms of the nomenclature, it's by convention that we use this specific type of system. So in this configuration, messenger RNA is said to be in the plus configuration. And the complementary to the messenger RNA is considered to be in the negative conformation. So positive strand RNA viruses, these are single-stranded RNA viruses, and they have the same orientation as that messenger RNA. So as soon as positive strand, single-stranded RNA viruses enter the cell, that RNA, because of the polarity of it, the plus strand is in the same conformation as messenger RNA, and that allows the virus to start right away in terms of producing proteins, specifically the RNA-dependent RNA polymerase protein, to allow it to manufacture negative strands, which allows it to manufacture more positive strands, and the whole cycle keeps on going. Negative stranded RNA viruses, they have to bring in that little piece of RNA-dependent RNA polymerase in order for it to function. Retroviruses are different. You will notice that the virus that causes AIDS, HIV virus, has two identical copies of its viral genome. Specifically, it's single-stranded RNA in the plus configuration. The problem is that that specific type of RNA doesn't mimic messenger RNA. Instead, that, as we'll show later on, uses an enzyme called reverse transcriptase, which is an RNA-dependent DNA polymerase, to actually manufacture double-stranded DNA from the single-stranded RNA genome of the virus. That double-stranded DNA now integrates into the host DNA itself. More on that when we get to the HIV section. In terms of what is actually being produced, so this is more of a summary. So the early stage proteins are synthesized soon after infection. So as soon as that, and we take the simplest case, as soon as that coronavirus gets into the cell, remember coronaviruses that's going around right now is essentially a single-stranded plus configuration RNA virus. As soon as it enters the cell, it's going to manufacture early phase proteins. And these proteins tend to be proteins that are necessary for the viral survival. They tend to be the RNA-dependent RNA polymerase, as well as proteins that can use to combat our innate system. And we'll see examples of that one, especially when we get to the HIV virus. So these are necessary for replication of virus nucleic acids. And they tend to act catalytically, in other words, the building, and they're synthesized usually in smaller amounts. Late phase proteins are synthesized later, and these usually include proteins that are involved with the virus coat. In other words, they're structural components. And these are usually synthesized in way more quantities, very large amounts. And the more that the virus can produce essentially means that the more assembly, the actual virions can occur, and more viruses that will actually leave the host itself. Okay. Now we're going to get into an overview of the actual bacterial viruses and animal viruses. First, we're going to have an overview of bacterial viruses, and then we're going to get into some of the animal viruses. And finally, we will talk briefly about the retroviruses, because the last lecture that we'll have later on will be exclusive towards retroviruses. I told you earlier about this bacterial phage and how it enters a bacterium, specifically E. coli. Let's look at that life cycle. It's a very, very short life cycle. This virus has to have complete control of the cell by the time the bacterial divides. And the life cycle of this bacterium is about 25 minutes, which is phenomenal. So there's a huge amount of energy being devoted to this type of production. So we have initial infection, followed by early phase proteins, messenger RNA that's coming in. So this is DNA coming into the cell, going to hijack the host cell machinery and essentially produce early RNA, which produces early proteins. Here we talk about mid-proteins, middle proteins. So we have mid-phase proteins being manufactured, and then we have late RNA followed by late proteins. We have assembly of the actual phage head and tail. So the head is spherical head, icosahedral, and here we have our helical tail. We have assembly of the virus, and in this particular case, we have the virus will actually lyse the cell. All this has to be done before the actual bacterial cell replicates itself, which is phenomenal in terms of how much time this virus goes through in order to infect the cell and eventually lyse the cell. At this stage, please look at the 2 videos that are accompanying this particular lecture. It's going to talk about virus replication of a bacteriophage in a bacterial host, and we call it a virulent bacteriophage. And that means that the host will be lysed.
Virulent phages carry out a simple life cycle called the lytic cycle. The phage attaches to the host cell during the attachment stage and then penetrates the host cell and injects its DNA during the penetration stage. After penetration, the phage causes the host DNA to break into small pieces. The phage then uses the host machinery to synthesize new copies of its DNA. This process is part of the biosynthesis stage. Biosynthesis also involves production of viral proteins. Once biosynthesis is complete, the phage components are assembled into virions during the maturation or assembly stage. In the release stage of the lytic cycle, the cell lyses, releasing the phage virions. These released phages go on to infect other cells.
The second video on viral replication in terms of a bacterial host is a temperate bacteriophage. This is where the actual virus integrates its genome into the host genome and is able to make more copies of itself. As the bacteria grows and divides, we're getting more and more and more viral genome being manufactured because it's integrated into the actual bacterial genome. As the bacteria grows, the virus genome will multiply as well. At some point, the virus can enter a lytic stage where the virus will lyse the cell and essentially liberate its virus. How does this happen? Well, at some point, that little piece of viral DNA that was integrated now comes out of the bacterial genome, and then the machinery, the host machinery of that bacteria are now hijacked. The virus produces more viral components, and eventually it will lyse the cell and liberate itself. So why does it go through all that? Well, one of the main reasons a temperate bacteriophage is to increase the numbers of. So bacteria grow very, very rapidly. And if the virus is associated with the genome of the bacterium, it's going to grow rapidly too. You're going to get more copies. So we're going to get a high copy number of the actual virus. So it's a survival strategy for that specific virus.
Temperate bacteriophages carry out 2 types of life cycle, the lytic cycle and the lysogenic cycle. The lytic cycle for temperate bacteriophages is similar to the lytic cycle for virulent phages. The phage attaches to the host cell during the attachment stage, and then penetrates the host cell and injects its DNA during the penetration stage. In temperate bacteriophages, the phage DNA forms a circle, which can either replicate and be transcribed to produce phage components in the lytic cycle, or can proceed to the lysogenic cycle. During the biosynthesis stage of the lytic cycle, the phage DNA directs the host cell to synthesize viral components. The phage components are assembled into virions during the maturation or assembly stage. In the release stage of the lytic cycle, the cell lyses, releasing the phage virions. These released phages can go on to infect other cells. Recall that temperate phages can proceed either to the lytic cycle or the lysogenic cycle shortly after penetration. In the lysogenic cycle, the phage DNA integrates within the bacterial chromosome by recombination. The inserted phage DNA is called a prophage. Most of the phage proteins are repressed by two repressor proteins that are products of the phage genes. Whenever the bacterium reproduces, the prophage is also copied. The prophage is excised from the host chromosome in a process called induction. Induction can occur spontaneously through recombination or some other genetic event, or through the action of UV light or certain chemicals. At this point, the phage may enter the lytic cycle.
In terms of animal viruses, the virus, the virion, enters the animal, but it's the entire virus that normally enters the actual animal itself. Unlike in bacteria, where the bacterial phage stays outside the host and injects its nucleic acid, in this case DNA, into the bacterium. We have a nucleus. Bacteria don't. So within us, the nucleus can be a site of replication for many animal viruses, but not all of them. Specifically, the RNA viruses tend not to do the nucleus trick. The DNA viruses tend to do that. Animal viruses, we contain all the known modes of viral infection. So the easiest way to understand this is to look at the Baltimore classification. Okay? And it'll explain everything in terms of the functionality of these viruses in terms of production of messenger RNA. Many viruses that exist in terms of animals are enveloped. And so they take a little piece of that membrane with them as they leave. This phospholipid bilayer membrane. This is just showing you the examples of two major classifications. So we have the DNA viruses and the RNA viruses. We have examples of non-enveloped and examples of enveloped viruses, DNA viruses, and the same for the RNA viruses, where we have non-enveloped as well as enveloped viruses. Continue with animal viruses. What are the consequences of these infections? So we've seen the infectious process, and we've seen how the viruses enter our cells. We've seen how they act. If it's DNA, they just hijack our host machinery and produce proteins from that perspective. We've seen how RNA viruses work. So what are some of the outcomes of a viral infection? Well, let's actually look at the figure. So we have persistent infections, we have latent infections, transformations, possible cell fusion. So why are they doing all this? Let's go to the figure. So in terms of we have our initial viral infection, we have a number of outcomes. One of them can be, and this will vary depending on the nature of that specific virus. So we have scenarios where we can have a persistent infection, where you have the slow release of the virus without cell death. So here we have a scenario where we have a cell that is not being lysed, that's continually producing very, very slowly amounts of virus. So that's what we call our persistent infection. Another avenue is right here, cell fusion. Some viruses can actually call the fusion of cells together. You know, from a cell perspective, that's not a big deal, because when you're looking at our cells. Our cells can fuse. And so from that perspective, it's something they can do. And a good example is skeletal muscle. We have satellite cells that are located near skeletal muscle, and they can actually fuse with normal skeletal muscle cells to have a larger muscle cell. So cell fusion can happen. But why does it happen in terms of a virus? We're not 100% certain about that, but it can be related to what we call genomic stability. In other words, we're destabilizing the genome of that particular cell. Whether or not this will lead to consequences down the road is still under investigation. So in other words, this could be one pathway that the virus can destabilize the host genome and potentially cause cancer down the road. We do know that some viruses can actually transform cells. And some of these viruses can actually transform these cells into neoplastic cells. And there's been a host of viruses that have been implicated to this. Hepatitis B viruses have been implicated to the formation of hepatocellular carcinoma. SV40 viruses has been associated potentially with non-Hodgkin's lymphoma. Epstein-Barr virus has been associated with Hodgkin's lymphoma. So there are many viruses that have that potential. Whether or not they actually do is still controversial at this point. There's still research going on, but it seems that some viruses can transform cells. One of the other ways is the virus into the host. We have a latent infection, and then the host can actually be lysed and the release of the actual virion particles. So we have death of the cell and the release of the virus. So that's another outcome that can happen as well. We'll look at this pathway, the modification of this pathway, when we look at the HIV virus that infects our cells, specifically cells that include dendritic cells as well as T4 helper cells. That'll be in the final lecture. We've now covered the life cycle of a virus. Now we have a scenario where the virus is now fully assembled, which is a very complex process, and we have the release of the virion particles. So this example shows the process of budding in envelope viruses. So at the surface, we have essentially what we call a lipid raft, where we have an accumulation of lipids as well as glycoproteins, huge numbers of viral glycoproteins. They accumulate within a specific area. And this viral capsid is now directed to that lipid raft by our host machinery, and we have budding of the virus out of the host until it's fully released. A good strategy is to stop this from being released at all. And that's when we look at the HIV virus. This is one of the ways that this virus, we can actually control this virus. So if this was the HIV virus, what can happen is we have a specific protein within us called tetherin. Tetherin is a membrane-bound protein and actually tethers the virus particle to itself, so it's not being released. It then triggers endocytosis, brings it in, and then the virus protein complex is now degraded. Guess what? Well, that virus makes proteins to counteract tetherin. And so all of this is a tit-for-tat relationship we've been talking about all year round. So this showing you, again, the escape of the virus from the host. In this particular case, the virus has a capsid, nucleic acid, as well as the envelope, virion with its spike proteins that will go off to infect other cells. At this point, we're going to give you a very brief introduction to retroviruses. And we will give you more depth of what it is and a more depth in terms of the viral genome of the HIV virus in the next lecture. So retroviruses, these are RNA viruses that replicate through a DNA intermediate. They are envelope viruses. They contain within the virion itself They contain reverse transcriptase, as well as other proteins that are necessary for its survival. In other words, integrases as well as proteases. And oddly enough, it requires transfer RNA from the donor. It doesn't make its own transfer RNA. It uses your transfer RNA in order to help initiate its replication. It needs that. And we'll see that in the next video. This is just showing you an overall look of the HIV virus. We have 2 identical copies of the single-stranded plus strand RNA, as well as other components that are necessary for the survival of this virus. So within the actual viral virion particle, you have to have copies of reverse transcriptase, integrase, proteinase, in order for this virus to be viable. If these are not present, then this virion will not be functional. What's the main reason for that? One of the main reasons is we don't make this type of reverse transcriptase. So what is reverse transcriptase? It's an RNA-dependent DNA polymerase. We don't manufacture that, although our telomerase is actually that type of protein, but it's not functional in terms of this. Continuing on with retroviruses. So they have a unique genome. All retroviruses, for some reason, have 2 identical copies of this single-stranded RNA. This could be energetically favorable in terms of how this virus actually functions. It could be a way of, maybe I'll leave all that till we get to the next lecture, then we can talk about that. It does contain specific genes. We have the GAG proteins, which encounter, these are the structural proteins that are manufactured. The polymerase pol protein, which encodes the reverse transcriptase and integrase, and then we have our ENV proteins, which basically make the envelope proteins. In terms of the genome structure, I'm going to show you a much more detailed version in the next lecture, but just understand that these proteins are arranged in a specific order found within the actual genome itself. So this genome, because it has this coding sequence here and this coding sequence here, doesn't behave as messenger RNA. These are sites that the RNA polymerases essentially lock onto. Process of replication. You will see that the sequence of events is basically the same to all viruses. We have entrance into cell, removal of the virion envelope at the membrane. The proteins that it comes with it start to work. Okay, so we have reverse transcriptase making essentially this into DNA, and then the integration of this retroviral DNA goes into our host cell. And then as time goes on, we have transcription of these retroviral DNA to manufacture specific host proteins, viral proteins, and from there we will see how this works out within the next lecture. And this figure just summarizes the HIV life cycle. So we have the virion located up here. It enters the cell, uncoats the single-stranded plus configuration RNA, interacts with reverse transcriptase to make double-stranded DNA. This goes integrated into your host DNA, where it can manufacture viral messenger RNA and more copies of itself too. Because it's in the plus configuration, you're going to see a lot of manufacturing of this particular strand, and this will produce many, many, many copies of single-stranded plus configuration that will form along with the nucleotide proteins to form the nucleic capsid, and this virus will eventually bud out into releasing of more virion particles. That's a very, very simplistic view of the HIV virus. Next lecture we shall show you a video on how this happens. And in addition, for the next phase of this course, I would like you to watch the David Suzuki special on host microbial interactions. It's called The Antibiotic Hunters. It's a very good program that shows you the current state of antibiotics within our system. Thank you very much, and the next lecture after that will be patient 0, and that's where we will talk about the origin of the HIV virus. Have a good day.